Right, I think we should get started. Hello everybody, and welcome to our biology seminar. Today, it is my distinct honor to welcome Ken Irvine, who is currently a distinguished professor and the interim director of the Waxman Institute of Microbiology at Rutgers University. Dr. Irvine got his undergraduate degree in chemistry from Williams College here in Massachusetts, and a PhD in biochemistry from Stanford, and where he worked in the lab of David Hognes. My understanding is that he started working with Drosophila at that time and has kept this passion for fruit flies to this day. And then he carried out postdoctoral research at Princeton University with the Nobel laureate Eric Wieschaus. Dr. Irvine's expertise and main area of research is in the regulation of organ growth and tissue patterning by conserved developmental regulators. And in particular, he made key discoveries of how Notch signaling pathway is controlled by its ligands and post-translational modifications such as glycosylation. And more recently, Dr. Irvine's research focused on the Hippo signaling pathway and its role in growth control. And in particular, how this pathway is connected to mechanical forces in the developing organs. So Dr. Irvine was awarded several prestigious fellowships at various stages of his career and was an investigator of the Howard Hughes Medical Institute. He published over 100 peer-reviewed research papers and reviews, and plays a very active role in the Drosophila community, organizing conferences and participating in various ways. And so without further ado, I will give the floor to him. The title of his talk today, Biomechanical Regulation of Organ Growth. Dr. Irvine.
Okay, thank you very much for that kind introduction, and thank you for the virtual invitation to visit, and hope to be able to visit in person someday. So I'm going to restart the screen sharing here. Does that work out? Can everyone see the screen?
Yes.
And I should say I don't mind being interrupted if anybody wants to try to yell out a question in the middle of the talk. So, oops, hang on a sec. Let me just get a laser pointer. So to begin with, I'm really a developmental biologist, and the kind of questions that we've been working on for a long time relate in most cases to the questions of trying to understand how the sizes, shapes, and proportions of different body parts are controlled during development. And that question is illustrated very beautifully in this classic drawing from Leonardo da Vinci, the Vitruvian Man, which is supposed to show the perfect proportions of the human body. Now, we don't work with humans for a variety of reasons, but what we work with instead is Drosophila, as Yksi mentioned, which also has beautifully proportioned appendages that can be studied. And so this is a great model for investigating questions of how the sizes and shapes of different parts of the body are controlled. And we know from decades of research that at a molecular level, the types of molecules that are involved in controlling size and shape of organs are very highly conserved, all the way from flies to humans. So one of our favorite model organs in the fly, and the one that will be the focus of most I want to tell you about today, is the developing Drosophila wing, which is schematized in cartoon form here. And this wing of the fly develops from a cluster of undifferentiated epithelial cells called a wing imaginal disc, which is shown in cartoon form here. And it's actually just this region shaded in green here in the developing wing disc, which is going to give rise to the blade of the adult wing. And I also just want to point out, just so you can sort of visualize the transformation between them, because we're ultimately interested in how the size and shape of the wing is controlled. But most of our work, since we're studying how this is regulated during development, is actually focused on studying wing imaginal discs. During metamorphosis, this wing imaginal disc region, the wing region, is essentially going to extend outwards, if you imagine the disc turned on its side, and then flatten. So in the adult wing, you have two cell layers, a dorsal layer and a ventral layer, which come from the two halves of the wing disc. And the center of this developing wing is going to end up being the edge of the adult fly wing. So this shows you some pictures of wing imaginal discs just with a nuclear stain at an early stage of larval development and at a late stage of larval development. And one of the reasons that the wing disc has been a very nice model for studying growth control is it does increase in size about 1,000-fold during the larval stages. So growing from about 50 cells to about 50,000 cells. And, if you want to understand, how is this growth controlled? We know that cells in this developing organ are receiving a variety of biochemical cues that give them information about things like the developmental stage or the age, contacts with neighboring cells. There are various secreted growth factors like Wnt and BMPs that tell cells information about their position and influence the growth of this tissue. The metabolism and nutritional status of the organism is, of course, also very important. But in addition to these biochemical cues, cells in a developing organ like the wing also receive mechanical cues because they experience mechanical forces through the contacts with neighboring cells and extracellular matrix. And those mechanical cues can also influence cell fate decisions and cell proliferation decisions. And that's mostly what I want to talk to you about today. So I think intuitively, most of you are familiar with the concept, if you think about it, that mechanical forces that tissues in our body experience can influence growth, because we can see this if you look around you. The skin, proliferation of cells in your skin can be stimulated by stretching, and that has cosmetic applications, also health applications. For example, if someone was obese and then loses a lot of weight. Obviously, bodybuilding, any kind of growth of the muscle is in part a response to a repeated mechanical strain in that tissue. Your bone as well, growth of the bone depends upon the mechanical strain that the bone experiences. That's illustrated in the example shown here, which is as a nice internal control, because it's the left and right arm of a professional tennis player, and the playing arm has a higher bone mass than the non-playing arm. So we're interested in the question of, what are molecular mechanisms by which mechanical forces that cells experience can influence things like cell growth and cell proliferation. And of course, there are going to be many answers to this question, but I'll be telling you today about one answer that applies in some epithelial tissues. And that answer, that mechanism I'll tell you about, is a mechanism that works through regulation of the Hippo Signaling pathway, which is probably familiar to many of you through the work of your colleagues like Yksi and Jens, but I'll give you a brief overview of it with this slide. So Hippo Signaling is a signal transduction network where there are upstream cues that regulate the activity of kinases. In the Hippo pathway, the key kinases are called Hippo in Warts, and then these kinases regulate the activity of a transcription factor, which in Drosophila is called Yorkie. And it turns out that amongst the key targets of this Yorkie transcription factor in many tissues are genes that play important roles in promoting growth and cell cycle progression and inhibiting apoptosis. And the way the pathway was first discovered is through mutations, some of these upstream genes that function as negative regulators of Yorkie. So when they're mutant, you can get overgrowth. And an example of that is shown here, where you have an adult fly that has a mutation in a gene called Warts in a patch of cells on the back of the fly. And even if you don't work with flies, I hope you can tell this is not normal. This is a growth, and maybe you can even imagine why the gene got its name, but there's a sort of tumorous growth that's composed of Warts mutant cells that's growing out of the back of this fly. Now, Warts is a kinase that negatively regulates this Yorkie transcription factor by phosphorylating, and that phosphorylation promotes its cytoplasmic localization. And you can see an example of that in the picture shown in the bottom right here, where you're looking at a close-up of some cells in a developing wing. And at this stage in the wild-type cells, the protein is mostly cytoplasmic. So you see this honeycomb pattern because it's not in the nucleus. But then there's some patches of cells here that are mutant for Warts. And in those Warts mutant cells, the protein has gone into the nucleus. Now, this Hippo pathway is highly conserved. I usually tell people that it's very highly conserved except for the names of all the genes. So I apologize for that. But in terms of the actual sequences and structures of the proteins and their biochemical relationships and their functions, there's a lot of conservation between Drosophila and mammals in this Hippo pathway. And the functional conservation is illustrated on the bottom, where in the bottom left, you have a normal wing imaginal disc, and then at the same scale, a wing imaginal disc in which the Yorkie protein has been overexpressed, resulting in high activation of Yorkie. And on the bottom right, you have a mouse liver, and then a mouse liver in which one of the homologs of Yorkie protein called YAP has been overexpressed, and that's stimulated an overgrowth of the liver. And this is first shown from some work from DJ Pan's group. And because this pathway does have important effects on growth and also cell-fate decisions, it might not surprise you to know that the Hippo pathway has also turned out to be very important in a lot of cancers. So you see in many different, particularly solid tumors, elevated levels in nuclear localization of the transcription factors at the end of the Hippo pathway, which in mammals are called YAP and TAZ. And an example of that is shown here in a mouse lung cancer model where lung cancer has been induced by activation of KRAS. And these tumors that were formed are mostly associated with elevated levels of nuclear YAP protein. And in fact, in human non-small cell lung cancers, about 65% of the time, people see elevated levels of nuclear YAP. Okay, so I want to get back to talking about the mechanics. And let me just first say with this cartoon, if we think about, what is the mechanical environment that cells experience, you can think about different aspects of the mechanical environment. So cells, for example, have mechanosensory channels or cilia that respond to forces cells experience, maybe shear flow from fluids. Cells have attachments to an extracellular matrix, and they can experience forces through these attachments to extracellular matrix, for example, respond to differences in stiffness of the matrix or stretching of the matrix. And in addition, cells have contacts to neighboring cells through a structure called the adherence junctions, and they can experience forces through these adherence junctions. And both adherence junctions and the contacts to the extracellular matrix, the focal adhesions, are connected to the actin cytoskeleton, and that provides a way for cells to sense and respond to force. And for today's talk, our focus has been on forces that cells experience at these adherence junctions. And the adherence junctions can be labeled with a GFP labeled e-coherent. So what you're looking at down here is a wing imaginal disc in a live ex vivo culture. Cells are proliferating. That's indicated by the red staining, which is just a protein that binds to microtubules. And then the cells are outlined by this e-coherent GFP. In the bottom right now, I'm showing you a close-up of some cells in a wing disc. They're outlined again by this e-coherent GFP. And what we've done in this little movie that's looping here is take a pulsed laser and sever the cytoskeleton along one of the cell-cell junctions. And when you do that, what happens is that you can see the neighboring vertices spring apart. And this is a type of assay that people use to show that these junctions are under tension. So imagine, for example, if you took a rubber band and stretched it and then you cut in the middle, the free ends would spring apart. And that's essentially what's happening here. And in fact, if everything else is equal, you expect that the velocity of the recoil after you cut this is going to be proportional to the amount of tension on the junction. So then the key point for my talk today is that for most of wing development, these junctions in the developing wing are under tension as indicated by this type of assay. So for the first part of my talk, I want to review some work that was published several years ago. And so I'm going to give you the take-home message first and then just show a few of the key observations. And this work led us to propose a model for a biomechanical signaling pathway that we call the Jub pathway, or the Jub Biomechanical Pathway, which is a mechanism through which tension in the actin cytoskeleton can regulate the Hippo pathway. And the essence of this mechanism is that under conditions of high cytoskeletal tension, the Warts kinase forms a complex with an inhibitor called Jub at adherence junctions. These proteins are recruited in a complex at adherence junctions, and Jub functions to inhibit this Warts kinase. And consequently, since this Warts kinase is inhibited, it cannot phosphorylate the Yorkie transcription factor and inactivate it. So instead, the unphosphorylated Yorkie can go into the nucleus and regulate downstream gene expression. Under conditions of low cytoskeletal tension, you don't form this inhibitory complex, so the Warts protein can instead interact with other proteins in the cell, including proteins that are involved in its activation, like expanded in Hippo and Salvador. They promote activation of Warts, which involves its phosphorylation, and then Warts phosphorylates and inactivates the Yorkie protein. Let me, before I go on, just try to give you a sense of what we think this pathway contributes or can contribute to growth control in the wing. And that's indicated by comparing the sizes of wings. In the middle, to a situation where we have essentially no Jub. So Jub has been knocked down in the developing wing using RNAi, and you can see that you get a much smaller wing. And on the far right is a case that I'll explain more in a few minutes, which we think of as activated Jub, where Jub is forced or allowed to go to these junctions, even in the absence of high tension, and that can give you a larger wing. So you can have some, very obvious effects on the growth of the wing, but I do want to point out that you still get a wing even if you don't have detectable levels of Jub. So this is one pathway that controls the growth of the wing, but certainly not the only pathway. So I next want to point out that changing tension in the actin cytoskeleton in the developing wing can have effects on the Hippo pathway, and also introduce you to a type of assay that we use a lot. And that assay is to express a transgene of interest in one half of the developing wing. So in the example shown here in the cells labeled in green, and that lets us use the other half of the developing wing as an internal control. What we're looking at here in magenta is a reporter for the transcriptional activity of Yorkie that we use a lot. It's a transcriptional reporter for the expanded gene, which is a direct target of Yorkie. And in the top experiment here, we're just expressing GFP, and you can see the expanded expression is similar in the two halves of the disc. In the disc shown in the middle here, we've decreased cytoskeletal tension. We can do this in different ways, but in this case, this is using an RNAi against a protein called Rho-associated protein kinase, which is one of the key activators of myosin activity in flies. And that results in lower cytoskeletal tension in these cells, and that's associated with reduced expression of this Yorkie target gene, and then also other measures of Yorkie activity. And conversely, we can increase cytoskeletal tension. The experiment shown in the bottom down here, we've actually expressed an activated form of this kinase that increases cytoskeletal tension, and that's associated in the middle of this developing wing with actually increased activity of Yorkie, which you see here as increased expression of this transcriptional reporter. So one of the things that we did after we discovered this is to try to figure out, well, what other genes in the Hippo pathway might be required for changes in cytoskeletal tension or myosin activity to affect Yorkie activity. And those experiments led us to focus on this gene called Jub, which had already been identified as a protein that acts as an inhibitor of the Warts kinase and can physically interact with the Warts protein. And what we found, so the regulation, this inhibition of Warts by Jub means that it's effectively a positive regulator of Yorkie, since Warts inhibits Yorkie. So when you knock Jub down, you get reduced Yorkie activity, shown as reduced expression of this transcriptional target. And what we found is that this reduction of Yorkie was sufficient to completely block or suppress the ability of increases in myosin activity to increase Yorkie activity. So when you combine those two together, basically you see the Jub phenotype. It's a type of epistasis experiment, which suggests that cells actually need the Jub protein in order for increases in myosin activity to increase Yorkie activity. Now, another key result that got us very excited about this Jub protein is the observation that its subcellular localization is actually controlled by cytoskeletal tension. So using a GFP fusion to Jub, we can see that this protein normally localizes to the adherence junctions in cells, overlaps e-cadherin. But if we look in cells where cytoskeletal tension has been reduced, then we have lower levels of this Jub protein on junctions. And if we look in cells where cytoskeletal tension has been increased, then we have higher levels of this Jub protein on junctions. And we know from other experiments like Western blotting that the total amount of Jub in the cell is not changed by these experiments. We're just changing its localization. So that's very exciting, was very exciting to us to see that this protein that's genetically required for myosin to regulate Yorkie activity is in fact itself regulated by tension in the actin cytoskeleton. And then the final piece of data that I want to show you for this part of the talk is that we discovered that the Jub protein is actually responsible for recruiting the Warts protein to adherence junctions. So the Jub protein is at the junctions overlapping e-cadherin. It has a more punctate distribution. We actually think these puncta represent points on the junctions that are experiencing higher tension. But the key point for now is just to emphasize that the Warts protein also is found in puncta on junctions, and the Jub puncta and the Warts puncta overlap. And in addition, we can see if we make patches of cells that are mutant for the Jub gene, what we call Jub mutant clones, within those clones, you lose the localization of the Warts protein on junctions. So these types of observations and some others that I won't go through gave us this model for tension-dependent regulation of the Hippo pathway, which is based on forming this complex between the Warts kinase and its inhibitor Jub at adherence junctions experiencing high tension. So what I want to try to do for the rest of my talk today is to go through some of the follow-up experiments we've done in this in 3 different areas. So one thing that we're very interested in is to try to understand what's actually the mechanism. How could tension in the actin cytoskeleton recruit this Jub protein to adherence junctions? We were interested in the conservation of this pathway in mammalian cells. And most interesting to me as a developmental biologist is, what really is the biological function of this pathway? What role might it serve during normal development or physiology of the animal? So in terms of the mechanism, we really borrowed from work that had already been done looking at the interaction between another protein called Vinculin and adherence junctions, and work first from Yonemura et al., a group in Japan, the Riken Institute, had led to a model being proposed that alpha-catenin undergoes a tension-dependent conformational change that allows it to bind to Vinculin. So the observation is that Vinculin will go to adherence junctions under tension. It associates with a particular region of alpha-catenin, and it seems that that region that it associates with is accessible under conditions of high cytoskeletal tension, but not low cytoskeletal tension. And the idea is that alpha-catenin itself undergoes a conformational change because one end of alpha-catenin is essentially anchored in place. It binds to beta-catenin, which binds to E-cadherin, and the other end of alpha-catenin associates with the actin cytoskeleton. So if there's tension on the actin cytoskeleton, it can actually directly pull on this alpha-catenin molecule and expose a Vinculin binding site. So we thought that this model would also work well for thinking about or explaining how Jub might exhibit a tension-dependent localization to adherence junctions. And in fact, we could see that, of course, that if we knock down alpha-catenin, then we lose the localization of Jub to junctions. We could co-immunoprecipitate Jub and alpha-catenin together from lysates of wing imaginal disc, which is what's shown in this plot here. And then we went on to actually try to map what region of alpha-catenin is responsible for interacting with the Jub protein. So shown at the top here are sort of 2 different ways of describing regions of alpha-catenin. It's been described as having these VH regions, which stands for Vinculin homology, regions of homology, actually to the Vinculin protein. And then below is a more structural depiction, which I can best explain by showing you crystal structure of alpha-catenin. This is actually a predicted structure of the Drosophila alpha-catenin, but it's essentially the same as what you would see if you looked at the actual solved structures of a mammalian alpha-catenin. And alpha-catenin is mostly a bunch of alpha helical bundles, 4 helix bundles. And so we can depict those in a linear array like this. So we did some co-immunoprecipitation experiments to map what region of alpha-catenin actually bound to the Jub protein. And I'm not going to go through all of that in detail, but the take-home point is that we found that this alpha helical bundle right here, which we refer to as N2, so in the second helical bundle in the N-terminus, is responsible for binding to the Jub protein. And that was quite an interesting observation for us because the Vinculin protein, which also has this tension-dependent recruitment to alpha-catenin, binds actually to a different region of alpha-catenin. It binds to this M1 alpha helical bundle. So 2 different proteins have this tension-dependent recruitment to alpha-catenin, but they actually bind to different regions of the protein. We went on and characterized some other deletions and different forms of alpha-catenin. And interestingly, we observed that deletions of some regions of alpha-catenin, in particular this M1 region, actually increase the binding of Jub to alpha-catenin. So that's illustrated first here in a co-immunoprecipitation experiment where with the full-length alpha-catenin, the signal that we get is actually barely above the background to the negative controls, but we can get a pretty good immunoprecipitation if we use this delta M1 mutant form of the protein. And so the concept here, the idea here is that in some way tension is changing the conformation of alpha-catenin to allow the Jub protein to bind. The simplest way to think about it is that there's a Jub binding site that's exposed in a high tension conformation, but not in a low tension conformation. and that this deletion in some way changes the conformation in a way that also exposes this Jub binding site. And we can see this increased ability of Jub to associate with alpha-catenin in vivo, as well as in these in vitro co-manifestation experiments. So an example of that is shown in this slide. So in the top is a control. Again, this is our experiment where we take a wing imaginal disc and we express transgenes of interest in one half, on the right half here. And the control at the top, what we've done is to get rid of the endogenous alpha-catenin using RNAi and then express an RNAi insensitive transgene. So the top just has wild-type alpha-catenin, and then we're monitoring Jub localization, and you see the normal Jub pattern. In the experiment on the bottom, instead of replacing endogenous alpha-catenin with a wild-type form, we replace it with this M1 mutant form. And as you can see, that actually increases the recruitment of the Jub protein to adherence junctions. So expressing this mutant form of alpha-catenin can give us a stronger recruitment of Jub. And in addition, it can actually give us at least partially tension-independent recruitment of Jub. Because if we do this experiment now in the flies in which we've also reduced cytoskeletal tension by using this RNAi against the Rho-associated kinase, normally, as I showed you before, that would reduce the recruitment of the Jub protein to junctions. But if we have this alpha-catenin mutant, then we still see the high recruitment of the Jub protein to junctions, indicating that this recruitment is tension independent. Now, this M1 region is the region that binds to vinculin, and in fact, in flies, we can see that vinculin protein is lost, but we don't think that's functionally relevant to the effects on Jub. And the reason I say that is that we know from looking at vinculin mutants or vinculin knockdown that it doesn't have any effect on recruitment of Jub. In fact, in flies, you don't even need vinculin at all. So vinculin null mutants make perfectly, apparently happy, healthy flies. Other thing that we observe now using this alpha-catenin mutant that constitutively recruits Jub is that it increases Yorkie activity. And that's the mutant I described a few minutes ago that gives us a bigger wing. And if we express it in one half of a wing disc, we can see, looking at readouts of Yorkie activity like this transcriptional target, expanded Yksi, that it's actually increased in cells that express the mutant form of alpha-catenin and compared to the control half of this tissue. And this is actually quite a significant result for us because our initial work was done by manipulating myosin activity, doing things like expressing RNAis against the Rho-associated protein kinase. But of course, changes in myosin activity could have had a lot of other types of effects besides their effects on Jub. But now, in a sense, we've uncoupled the changes in myosin activity from the recruitment of Jub, because we know that expressing this mutant form of alpha-catenin doesn't visibly change tensional on the junctions or distributions of myosins. All it does is to increase the recruitment of Jub, and that's sufficient to give us this increased Yorkie activity. So just to summarize this section of the talk, I want to point out that we feel that our results argue that alpha-catenin really is the mechanotransducer that's responsible for recruiting Jub to junctions under tension. And that's mainly because we can make mutant forms of alpha-catenin that alter its recruitment, and we can show that they directly bind to each other. Interestingly, Jub and vinculin have conceptual similarities in both being recruited to alpha-catenin under tension, but the way that they interact is very different. They associate with different regions of the protein, and there are deletions that can increase their recruitment that map to different regions of the protein. And also we can see that if you have an alpha-catenin mutation that increases Jub recruitment, that can be sufficient to increase Yorkie activity. So I want to spend a few minutes telling you a little bit about some work we did in mammalian cells looking at this pathway. And we don't necessarily always want to go to mammalian cells because we did the work in flies, but there are a couple reasons for doing this. The first is that there's been quite a bit of work in mammalian cells looking at how the cytoskeleton regulates the Hippo pathway. And most of the work that's been done has identified links between tension experienced at focal adhesions and Hippo signaling, whereas our work in Drosophila identified links between tension experienced at adherence junctions and the Hippo pathway. So we were curious, it wasn't clear whether this adherence junction pathway would also be found in mammalian cells. And the other reason is that there's just generally a greater complexity in mammalian cells. So in flies, we have a single member of this protein family called Jub. The main structural characteristic of these proteins is that they have 3 limb domains in their C-terminus. In mammals, you have 3 members of this family called a Juba, WTP, and limb D1, and we didn't know what level of redundancy there might be between them. So with this slide, I first wanted to show you that these three homologs of Jub actually all exhibit attention-dependent recruitment to adherence junctions in mammalian cells. That's shown by the experiment here. So when we work with the flies and we want to change tension in the cytoskeleton, we usually use some genetic tricks expressing some transgenes. When we work with the cultured mammalian cells, we usually give them drugs. So in this case, we've given them a drug called levastatin, which is an inhibitor of myosin activity, and that causes a loss of the recruitment of the LIMD1 protein to junctions. And for comparison, you can also see a loss of the recruitment of the Vinculin protein, which is what VCL is, to junctions. We could also see that the LATS kinase is recruited to junctions overlapping LIMD1. And again, I have to apologize for the differences in the names. I try to put up some sort of cheat schematics for you to help you make the connection. So LATS is the Warts homolog, which in flies gets recruited to junctions by the Jub protein. And we can see that actually the same thing happens in mammalian cells. The LATS protein is recruited to junctions, requires tension, and it overlaps with these Ajuba proteins. And then we did experiments to ask which of these three Ajuba proteins is actually responsible for recruiting the LATS kinases to junctions. And what we found, at least in the MCF10A cells, which is the main model that we've worked with, this is a human breast epithelial cell line, that it's specifically one of these three proteins is required, and that's the LIMD1 protein. So for example, if you knock down the Ajuba protein, we can show with antibody stains that the knockdown is effective, but the LATS protein is still on the junctions. Whereas if we knock down the LIMD1 protein, then we lose the protein from the junctions and instead it accumulates in the cytoplasm. And this has functional consequences as well that we can see looking at YAP activity. So in the conditions of culture shown here, this YAP protein is mainly nuclear in these cells. Knockdown of Ajuba has no detectable effect on YAP localization, but knockdown of LIMD1 tends to cause it to shift from a nuclear localization towards a more cytoplasmic localization. And you can also see changes in YAP activity in other ways, like looking at some of the transcriptional targets of the pathway. So this BERK3 and CDGF are two commonly used transcriptional readouts of Hippo signaling in mammalian cells, and their expression is reduced if you knock down YAP, and it's also reduced if you knock down LIMD1, but not other members of this Juba protein family. Okay, so I want to shift now to talk about function, and I'm going to talk first about function in a cultured cell model, and then we'll go back to the fly. And the function that I want to talk about is density-dependent regulation of cell proliferation. So if you've worked with cultured cells, you've probably heard of the phenomenon of contact inhibition of proliferation, which for most cell lines is really a misnomer because it's not contact inhibition, it's cell density-dependent inhibition. And the observation is that when you grow cells in culture at a low cell density, they proliferate quite well and happily, but as the cell density increases, their growth rates slow down. And that's illustrated in a schematic down here, which comes from a paper of a colleague of mine, Schraman, where he's plotted the relationship between the apical area of the cells, which is a readout of how dense they are, versus how long, what their doubling time is. And so you can see that they grow slower and slower as the apical areas change. Now, interestingly, the Hippo pathway has been known for a long time to be regulated by cell density. And that's illustrated by this figure that comes from this old paper from Kunlian Guan's lab, showing for two different cell lines, NIH3T3 and the MCF10A, that the YAP protein is nuclear under conditions of low cell density, but cytoplasmic under conditions of high cell density. And we got interested in the idea, the question of whether this density-dependent regulation, which was not fully understood, might have a biomechanical component, right? And in support of that idea, I want to show you with this slide that there are actually quite dramatic changes in the cytoskeleton when you compare cells cultured at a low density versus a high density. So these MCF10A cells at low cell density, we see nice actin stress fibers connected to adherence junctions, and they're coated with lots of phosphorylated myosin light chain. This is an indication that of myosin activity. So the phosphorylated myosin light chain is the active form of myosin. And conversely, under high cell density, much, much lower levels of phosphorylated myosin light chain, lower tension, and that's associated with, as you would expect, reduced recruitment of this LIMD1 protein to junctions, and also reduced recruitment of the vinculin protein to junctions. So to try to understand whether changes in cell density might be affecting YAP activity through changes in cytoskeletal tension and recruitment of these Jub proteins, we tried to uncouple the effects on the cytoskeleton from the cell density. And one way we did that is to take cells cultured at high density and then artificially activate cytoskeletal tension by using, in the example shown here, a drug called Rho activator 2. So these cells are at the same high density as the control cells shown above, but because of the Rho activator 2, they've got dramatic increase in activation of myosin, which you can see with this phosphorylated myosin light chain staining. And that's caused an increased recruitment of this LIMD1 protein to the junctions of these cells, and also caused an increased recruitment of the LATS kinase to these junctions, and an increase in YAP activity. So it shifts from the predominantly cytoplasmic localization that you would normally see at high cell density to predominantly nuclear when you treat with this Rho activator. Now, if this changes in LATS localization and YAP activity that we see when we activate tension in these cells depend on LIMD1, well, we can test that just by asking what happens when we knock down LIMD1. And what we see is that the recruitment of the LATS kinase and the increases in YAP activity actually depend upon this LIMD1 protein because they're suppressed when we knock it down with LIMD1 siRNA, whereas in control cells or cells where we've knocked down one of the other members of this family, we still see the junctional localization of LATS and the nuclear localization of YAP and activation of YAP target genes. So just to summarize what I told you the last few minutes, we see a conservation of this pathway that we first discovered in Drosophila in mammalian cells, where there's a recruitment of a member of this family. In mammals, it's LIMD1 to adherence junctions. And when it's recruited there, it can recruit this LATS kinase, and that's associated with increased activation of YAP, presumably because this kinase has been inhibited. And this can happen under conditions of high cytoskeletal tension, either by using a drug like Rho activator to increase tension, but also under conditions of low cell density. And conversely, under conditions of high cell density, tension in the cytoskeleton is lower, and then we don't form this inhibitory complex between LIMD1 and LATS at junctions, and so the YAP protein can be phosphorylated and kept in the cytoplasm. And so we discovered that this pathway can contribute to density-dependent control of cell proliferation. I do want to point out that this isn't necessarily true in every cell line. In fact, we see this very nicely in MCF10A cells, but there's some other cell lines that we've looked at where LIMD1 does not seem to be required for cell density-dependent regulation of YAP. Okay, I want to go back in vivo now to the fly, and let me introduce this section of my talk by going back to the wing imaginal disc and making a couple observations. If you compare a younger wing imaginal disc, which is shown on the left here, to an older wing imaginal disc, one striking difference is that there's a great difference in the rates of proliferation. So cells are actually proliferating much faster at younger stages of wing development than they are at older stages of wing development. Another interesting difference is that there's a big difference in cell density. So at a younger stage, the cells have a more almost cuboidal shape. So this is a pseudostratified epithelium, but the apical areas of the cells are much larger at younger stages. As the disc grows, the cells get more and more tightly packed, and they actually become more and more columnar. So this is all at the same scale. You can see the epithelium becomes much, much thicker, and you get these very narrow, tall cells, very densely packed at the older stages. So this is obviously an interesting correlation, and we decided to look into this in more detail and do some more quantitative studies comparing cell density and how that affects tension along junctions and the Hippo pathway. So this first just shows you some confocal images from carefully staged discs throughout the key stages of larval growth. This is essentially throughout the third larval instar. And then in this heat map, I'm showing you some quantitation. So the top images show you just quantitation of relative cell areas in a heat map where the blue is a smaller cell area and red is a bigger cell area. And this, there's 2 points I want to make from this. One, it reproduces what I just told you, that in general, as you go from a younger stage to an older stage, the cells get more densely packed. So you see that as a reduced apical cell area. I do want to point out that there's a spatial pattern as well as a temporal pattern. So if we look in the developing wing, which is what's shown here, the cells become more densely packed in the center of this tissue, but not so much out on the periphery. Now, on the bottom here is a quantitation of the amount of myosin protein and Jub protein along cell-cell junctions in different stages, and these represent averages from several discs. But what they show you quite nicely, I think, is that there's a pretty good correlation between the amount of these proteins on junctions and the area of the cell. So at younger stages, you have more of these proteins on junctions. As the discs get older and older, you get less and less of these proteins on junctions, particularly in the center of the developing wing, not so much at the periphery. And then there's one exceptional region, which is a line right along the center of the wing, which is a place where there's a stripe of notch activation that alters the response. And I don't want to talk more about that for now. But aside from that stripe of notch activation, there's a good correlation between the areas of the cells, the amount of myosin on junctions, which generally correlates very well with junctional tension, and the amount of Jub protein that's recruited to junctions, which also depends on junctional tension. And that correlation also carries on if we look at the localization of the Yorkie protein, which is a proxy, a good proxy in most cases, for the activity of Yorkie. It's predominantly nuclear in the younger wing discs. As the discs age, it becomes more and more cytoplasmic. So if you look at these older wing discs, you see that it's predominantly cytoplasmic in the center region of the wing disc. And there's also pretty good, at least some correlation with patterns of cell proliferation. And again, we can quantify that looking at averages from several wing discs. You see the predominantly nuclear Yorkie in the younger stages, predominantly new cytoplasmic in the center, but not so much at the periphery or at this region where we have a stripe of notch activation. And some correlation with cell proliferation rates being higher in the younger stages. This is just based off intensity of EDU labeling, and reduced in the older stages in the center of the developing wing. So this correlation suggests that in fact this cell density dependent regulation of Yorkie through this Jub biomechanical pathway is playing a role in modulating temporal and spatial patterns of Yorkie activity during wing development and apparently affecting growth of the wing. To strengthen this link so that we're not just based on a correlation looking at different stages of wing development, we wanted to try to uncouple the cell density to some degree from the stage of development. And the way that we did that is to basically use genetic tricks to manipulate cell density. And I'll show you one set of experiments in which we transiently express an RNAi against CDK1, so cyclin-dependent kinase 1, and that suppresses cell division so that you can get a wing disc where the half of the disc where we've reduced the CDK1 levels has bigger cells, cells with not necessarily bigger, but cells with a different shape. So they have a bigger apical area, and you can see that displayed also in the heat map here. And if we look at these discs where we have the cells with a bigger area, we do in fact see higher levels of myosin on these junctions. And if we do these types of laser cutting assays that I introduced you towards the beginning of my talk, it actually shows us that there's actually higher tension along these junctions that have higher myosin. We can see higher levels of recruitment of the Jub protein to these junctions. And we can also see higher levels of Yorkie activity shown either by looking at the subcellular localization of the Yorkie protein, which is relatively more nuclear in the cells and where we have the bigger cells, and also higher levels of expression of this transcription report of the expanded LAC-C in cases where we make the bigger cells. So these observations, we think, strengthen the idea that there's a connection between the density of the cells during development and regulation of Yorkie activity through this biomechanical pathway. And the way we think about what this might be doing for the animal during development is that, one of the major questions in thinking about developmental regulation of growth is how do organs know when to stop growing? Why do they stop growing when they've reached their correct size? And part of an answer, at least we think, is that in some organs like the wing, as the tissue grows and it approaches its final size, the cells become more and more densely packed. And so that provides a signal to these cells that, okay, we've approached the final size now, we've reached the density of cells that we have in the mature organ, so it's time to stop slowing down the growth. So we think that is in fact a biological role for this biomechanical pathway. Okay, so in the last few minutes, I want to tell you a little bit of a twist on this Jub biomechanical story, some more recent experiments in which we found a role for this tension-dependent recruitment of the Jub pathway that actually is disconnected from its role in Hippo signaling. And we got interested in this idea because we found that this Jub protein is basically recruited to junctions under tension throughout Drosophila development, even in places where there's no apparent role for the Hippo pathway. And that's illustrated in these pictures here, where we're looking at either at a movie or some fixed images of Drosophila embryos, and you can see there are these patterns of Jub recruitment. This is actually in the ventral furrow. This is on the dorsal side of the embryo during a process called dorsal closure. And basically we see patterns of Jub recruitment which match very well to where we know there's higher levels of myosin activity. But there's no role for Hippo signaling. So we wondered, what is the Jub protein doing in these places? Is it doing anything? Why is it exhibiting this tension-dependent recruitment? And we ended up focusing on this process called dorsal closure, which if you're not familiar with it, it's been studied as a sort of model for wound healing in flies. And just to give you a visual or impression of this, this is a little movie showing process of dorsal closure with cells just outlined by e-coherent GFP. So there's this dorsal tissue called the amnion serosa where cells are undergoing apoptosis and being lost. And then there's these lateral epithelial cells, which are basically moving dorsally as the amnion serosa disappears. It turns out these amnion serosa cells are actually constricting and helping to pull this epithelium up towards the dorsal side of the embryo, where then meet the cells on the other side and seal up to form the final sealed epidermis of the embryo. So there's a lot of quite striking accumulation of Jub protein in these cells during this process. And it turns out that when we looked in animals that are missing Jub, so there's something called Jub germline clones, because you have to get rid of both the maternal and the zygotic Jub contributions. But when you do that, you find very interesting defects in the shapes and organization of these cells. So dorsal closure still happens. But if you look at the cells undergoing dorsal closure, they look very different. So on the left here is a control case. These cells have a sort of nicely arrayed parallelogram shape in this dorsal epidermis as they're migrating dorsally to seal up the embryo and the amnion serosa is disappearing. If you look at the same stage in the embryos lacking Jub, you can see, I hope, that the shapes and organizations of these cells look very abnormal. And one characteristic feature of this abnormality is that we sometimes see what we refer to as rosettes, and this is a place where 5 or more cells meet at one place. We have a vertex where 5 cells meet. This is something that is seen only very infrequently in the wild type case, and when you do see them in wild type case, they don't last very long, but you see many more of them, and they last longer in Jub mutant embryos. And that was really a diagnostic phenotype for us, because it turned out that there was, that's very similar to mutations or to embryos that are lacking the function of a protein in Drosophila called STEP-key or STEP, which is an RF-GEF, which is a GEF for R family G proteins. It's also known as a cytohesin. And work from Tony Harris's lab had described a role for this STEP protein during this dorsal closure process, and they get these rosettes of cells. And their work had led to a model being proposed that this STEP protein is playing an important role in a feedback loop that controls tension in this tissue and is basically needed to allow these cells to relax as they're being pulled by the contraction of the amnion serosa so that they can stretch out nicely and seal up and move and migrate in response to stretch and flatten in response to this stretch. And also some evidence that this STEP-key protein localization was being controlled by tension. So we had seen now a very similar phenotype in Jub, and whenever you have 2 genes with very similar phenotypes, you think that maybe they're working together. And since we knew that the Jub protein was recruited by tension, we got interested in the question of whether Jub might actually be responsible for recruiting the STEP protein to junctions under tension. To look at this, we went back to our wing disc model. And with the top set of pictures shown here, I want to point out that the Jub protein, which as I mentioned before, has this sort of punctate distribution on junctions. If we look at the Stepke protein, it has also a somewhat punctate distribution, which overlaps quite well with the Jub protein distribution. And more importantly, this localization of the Step protein to junctions depends upon the Jub protein, because if we get rid of the Jub protein in the experiment shown here using RNAi, then we lose the recruitment of the Step protein to junctions. And in addition, once we realized that we were seeing this effect in the wing discs, we looked more closely at cells in wing discs, and we realized that there's an effect of Jub on the organization of cells that we hadn't noticed before. So normally the epithelial cells in the wing, most of them are roughly hexagonal or close to hexagonal. But if you look in wing discs where we've depleted either the Jub protein or the Step protein, you find that the organization of the cells is much more abnormal. You have much more variety in shapes and sizes of cells that look very irregular. And in addition, you have much more irregularity in the distribution of myosin protein. So the myosin protein localization is shown here. It's not perfectly even, but you tend to see some myosin around all of the cell junctions at roughly similar levels. But when we look in wing discs where we've depleted either the Jub protein or the Step protein, the striking thing we see is a much more uneven distribution of the myosin protein. So some junctions have very high levels of myosin, and it tends to form these cables, intercellular cables. We have high levels of myosin, and other cell junctions have no detectable myosin at all. So in some way, this Step protein is needed to modulate the distribution of myosin in this epithelial tissue, and we think in that some way is playing an important role in organizing giving you even distributions of shapes of cells. So then the last point that I want to make is that this biochemical mechanism, which we think depends upon tension-dependent conformational changes in alpha-catenin, recruiting this Jub protein, is actually playing multiple roles in the cell. It has an effect on Hippo signaling through recruitment inhibition of this Warts kinase, but also has effects on the distribution of tension in the shapes of cells through recruitment of this Step protein, which is thought to ultimately be able to influence the activity of Rho through regulating Rho endocytosis. Okay, so I'm going to close just with a little summary of what I've tried to tell you about today. I told you first about this biomechanical signaling pathway that we discovered that involves a tension-dependent recruitment of the Jub protein to junctions under tension, where it can recruit and inhibit the Warts kinase. I told you about some of our evidence that this involves this is mediated through alpha-catenin, which can undergo tension-dependent conformational changes, which we think regulate the ability of the Jub protein to go to junctions. Told you about evidence that this pathway is conserved in mammalian cells, and that we think both in mammalian cells and in developing Drosophila wing discs, this pathway can play an important role in regulating Yorkie activity in response to changes in cell density. And lastly, that it also has effects on other processes, modulating cytoskeletal tension through this protein called Stepke. Okay, and then most importantly, I want to thank the members of my lab. So this is a picture of the lab from a few years ago in sort of happy pre-COVID days. I also took a more recent picture of one of our lab meetings. Everybody's actually still smiling, even though they're wearing masks. You just can't tell. And I particularly want to acknowledge Cordelia Rauskob, who did a lot of our early pioneering work on the Jub protein, and also some of the last work that I just told you about looking at the Step protein. Hervé Alago and Chris Markosian did a lot of our work on the structure of alpha-catenin and how Jub binds to the alpha-catenin protein. And Consuelo Ibar is responsible for a lot of work on the mammalian pathway and how this pathway responds to cell density in mammalian cells. So with that, I'm going to pause and be happy to take any questions from anyone. And I'm not sure if I should stop my screen share.
Probably.
Yeah, so let me.
So there's a big applause coming.
Okay.
And all right.
Okay.
Oh yes, great. Yes, so we can see.
Okay. I can only see two of you plus biology department. So
right. So I'm going to read. There are some questions in the Q&A already.
Okay, great.
So others, if you have questions, please type them up. So Heya Zhao, she's a graduate student in my lab, is asking, very interesting talk. cytoskeletal tension has been reported to play roles in Hippo pathway regulated cell differentiation. Does Jub affect tissue differentiation as well, other than proliferation and morphology? Have you tried to stain the wing disc of Jub RNAi with some sulfate markers like DPP?
Okay, yeah, so that's a great question. I mean, we haven't looked really into that specifically. I would just say in general that, Jub is affecting Yorkie activity. And certainly Yorkie does play important roles besides just controlling cell proliferation. I mean, that's sort of the first phenotype that was identified. It's a very dramatic phenotype, so we tend to focus on that. But there's lots of evidence now, both in flies and in mammals, that Yorkie or YAP proteins can affect cell differentiation or other cell behaviors. So I would expect that certainly if Yorkie's playing in a role in a tissue where Jub is active, that it could also be affecting these other readouts. But it's not something that we've looked at directly.
Great, thank you. Here's another question. This is from Adan Colon Carmona, faculty in our department. Is cytoskeletal tension all the same? In other words, are there differences between tension caused by actin cytoskeleton versus that caused by microtubule cytoskeleton? Is there interplay in the signaling via Jub, StepK, or something else?
Okay, yeah, that's also a great question. I should apologize because I tended to abbreviate and just say cytoskeletal tension, and I really meant actin cytoskeletal tension. So this Jub is not have any direct connection to the microtubule cytoskeleton. This recruitment depends upon this conformational change in alpha-catenin, which is specifically induced by tension in the actin cytoskeleton. So I think the only way that the microtubule cytoskeleton, there are contexts where there's crosstalk between microtubule and actin cytoskeleton, so that might provide a way for microtubule cytoskeleton to influence this pathway, but it would be an indirect effect. So really this pathway is just responding to tension in the actin cytoskeleton.
Okay, if I may, I have a question.
Yeah.
So I was curious about your CDK1 inhibition result. So you get bigger cells, but at least at the stage of the wing disc, it seemed that the general shape of the disc was okay. And my general question is about how much compensation can be built into the system. In other words, can you get a properly shaped organ with fewer cells if some of these mechanisms go awry?
Yeah, that's a great question, and the answer is yes. So we didn't do that, but there's some classic experiments that were done like a decade or so ago in flies where people did exactly that to try to block cell division and ask, well, can you make a normal sized wing with fewer larger cells? And the answer essentially is yes. So the mechanisms that are used to determine wing size can still operate even with cells of a different size.
All right. I actually had another question. So, are there any feedbacks from Yorkie back to the cytoskeleton? Right. So I think you primarily showed effects from upstream down to Yorkie, but there must be some controls, right? Yeah. That go back. Can you comment on this?
Yeah. So I'm sure there are feedbacks that go back, and there are studies more in mammalian systems, also I think in zebrafish, showing that Yorkie or YAP activity can regulate cytoskeletal regulators, so that there's definitely feedbacks where Yorkie and YAP control the cytoskeleton as well as being controlled by the cytoskeleton. This is I think every connection to the Hippo pathway is also subject to some kind of feedback regulation. I have to say that we until recently, we haven't really thought about it that much. We've actually started to think about it more because of this connection with the StepK protein, which clearly is modulating cytoskeletal tension, which could potentially, is modulating Jub recruitment. So then there's a kind of feedback there. StepK doesn't seem to have a big effect on the total amount of Jub, but it does affect the pattern of Jub because, Jub tends to get recruited to junctions in lines that match these myosin cables that you see. So you get a lot of Jub in some places and not so much in other places.
Great. Well, if there are no further questions, I want to thank Dr. Irvine again for joining our seminar, giving a fantastic talk. Thank you. There's thanks from Adan. Right after the talk, we'll probably take a 5-minute break. Okay. And let's get together for happy hour. I'm going to send a Zoom link right now. It wasn't already included in the announcement. I'm going to put it into chat here. Let's see if I can. Right. So I sent a link to all panelists again. So let's disconnect for 5 minutes. Would 5 minutes be enough, Ken?
Yeah, that's sure. That's great.
Yeah, and so we'll meet back at that address. Thank you very much again.
Great. Thank you.
Thank you.